Hi everyone, my name is John Moeller and I was recently awarded a PhD in microbiology from Emory University. Today in my World Microbe Forum Rapid Fire Talk, I'm excited to share with you my recent research using species-scale genomic analyses to understand the evolution of genes that influence phage host range in Staphylococcus aureus. The main question driving my bioinformatics survey is knowing phage host range genes characterized in Staph aureus and other species from a comprehensive literature review, how are these distributed in our database of over 40,000 curated Staph aureus genomes? Are there clades that are more phage resistant than others? Do phage resistance genotypes predict phenotype? And finally, are clades that gain antibiotic resistance or virulence genes less likely to have phage resistance genes or vice versa? It's known that phages are the main drivers of horizontal gene transfer in the species through a process called transduction. I also have a number of hypotheses about phage resistance evolution. In an arms race scenario, where phages and Staph aureus hosts are rapidly co-evolving, I'd expect there to be positive diversifying selection on protein-coding phage resistance genes, but not on their essential sites. On the other hand, if these genes are truly essential, or if they're essential in multiple environments where Staph aureus tends to grow, I'd expect there to be negative selection on these genes to retain their function. I also expect there to be genomic and phylogenetic modularity amongst these genes. Genomic modularity, as many of these are encoded in operons, they're all transcribed as the same mRNA. And even if they're not, genes with similar functions tend to be more likely to be clustered than those with different functions. As far as the phylogeny, it is known that restriction specificity, for example, correlates with clonal complex, one level of phylogenetic organization, and there could be higher or lower levels of phylogenetic organization that correlate with particular phage resistance genes or alleles. Finally, I hypothesize there to be some kind of pooling effect of phage defense systems. It could be clades with a single phage defense system will pool accessory antibiotic resistance or virulence genes and prevent other clades from acquiring them, or the opposite. They prevent the gain of these genes from other clades. Before I describe my results, I'm going to go through the genes that have been included in this survey. These belong to the 3 stages of phage infection where resistance has been characterized in the species: adsorption, biosynthesis, and assembly. I've included wall-teichoic acid biosynthesis genes, which are responsible for synthesizing the receptor and thus would contribute to sensitivity, as well as capsule biosynthesis that form a thick layer of polysaccharide that prevent phage adsorption. As far as biosynthesis genes, I've included not only restriction modification, abortive infection, superinfection immunity, and CRISPR systems that have all been previously identified in the species, but also those that haven't. Phage defense systems only reported in the past 3 years, such as retrons, cyclic binucleotide associated antiphage signaling systems, the Zorius system, which is a novel type of abortive infection system, and many more. Finally, for assembly, I have included the 3 hallmark pathways by which Staphylococcus aureus pathogenicity islands will parasitize helper phage replication: capsid remodeling, DNA packaging interference, and inhibition of helper phage late gene transcription. In these first results, I'm showing you the level of conservation of the genes included in this bioinformatics survey. The left plot shows the genes ranked from most conserved to least conserved and colored by the stage of phage infection at which they act. The first 60 or so genes are core genes found in all of our curated Staph aureus genomes, about 43,000. And these are mainly red. These are adsorption genes by far that constitute the core gene set. These are followed by intermediate frequency genes that trail down from core genes down to those that are not found at all, and these are mainly green. They're mainly biosynthesis genes that are intermediate frequency. I also show the data as a violin plot on the right, and this shows the same pattern. Adsorption genes are most conserved, followed by assembly, and then biosynthesis genes with a long trailing tail. I next looked at phylogenetic associations with phage resistance, and first correlated clonal complex with either levels of accessory genome or accessory phage resistance gene content. It's known that clonal complex correlates with restriction specificity, which then affects transduction of accessory genes in general. Indeed, according to an analysis of variance tests, there is a statistically significant overall difference of these CC subsets for both accessory genome content as well as just accessory phage resistance genes. I also looked at phylogenetic signal, and what that is the idea that more closely related strains have similar phenotypes or are more likely to have the same genes. I measured this as the number of changes on the phylogenetic tree to get a gene presence absence pattern to match that expected by the phylogeny. More changes means less phylogenetic signal, fewer changes means more phylogenetic signal. And by far, assembly genes had the lowest levels of phylogenetic signal, which makes sense given these are horizontally transferred. I know all of these have phylogenetic signal based on comparisons to a simulated data set that was permuted 999 times, the gene presence absence pattern. I also asked whether there was any genomic modularity. I asked whether for a strain encoding one non-core phage resistance gene, how many others are encoded of each particular category. By far what I found for each category, noted by these subheadings, is for strains encoding an adsorption gene, it was more likely to have another adsorption gene than genes of any other category. The same was true for biosynthesis and assembly. So phage resistance genes are encoded together, most often with those of the same category. I then asked whether I found evidence of an arms race dynamic. If there was an arms race dynamic, I'd expect core phage resistance genes to have higher diversity, lower functionality, and higher selection than core genes in general. Instead, I found evidence of the opposite. There were not statistically significant differences in nucleotide diversity, functionality measured through delta bit score, or selection between core genes and core phage resistance genes. I then looked for correlations between gene presence and phage resistance phenotype. The one I could see was that superinfection immunity genes, which repress the infection of a temperate phage corresponding to a prophage, correlated with temperate phage resistance. The same pattern was true of accessory genome content. The more prophages or more superinfection immunity genes present, the higher the level of accessory genome content. Thus, in conclusion, adsorption genes are most conserved, followed by assembly and biosynthesis genes. All non-core phage resistance genes have phylogenetic signal, but it's weakest for assembly resistance. Phage resistance genes are encoded together and most often with those of the same type. There does not seem to be support for an arms race dynamic, and amongst the different categories of host range or resistance genes I studied, only superinfection immunity correlated with temperate phage resistance, not virulent phage resistance, as well as accessory genome content. And with that, I'd like to thank everyone who helped with the project, everyone in the lab for helping with phenotyping, phage resistance, with bioinformatics, and for general comments and criticism, those who provided reagents that were necessary, my dissertation committee for their comments and criticism, the National Science Foundation Graduate Research Fellowship Program for financial support, and all the phages for their hard work on the project. And with that, I appreciate your attention, and I'm happy to take your questions.